Hello everyone. Today we'll be doing structure of cilia and flagella. Okay, we'll see structure of cilia and flagella. Structure of cilia and flagella. So usually many times students have told me that ma'am, actually we get confused with the structure of this cilia and flagella. See, cilia and flagella, both of their structure is same. No need of any kind of confusions. Where you will get confused is when you study the structure of cilia flagella and then when you study the structure of centrioles, then you people will get confused. That is for sure. But I hope there will be no confusions after this class. Okay. Today what we'll do is structure of cilia and flagella. So before that we need to understand what is cilia and what is flagella. See, these are nothing but, what are this? These are nothing but hair-like outgrowths. Hair-like outgrowths. They are nothing but hair-like outgrowths. You know, the cells, usually cells, even if it is prokaryotic cell or eukaryotic cell, if you see, they will have some kind of hair-like outgrowths. But particularly we are talking about cilia and flagella in case of eukaryotes, not prokaryotes. Okay. So here, what we will see actually, what we'll see is that in cells, for example, if you take paramecium, if you take, you people know very well about paramecium, right? So somewhat it looks like this, not very perfect diagram, anyways. Okay, it looks like this. It looks like this, and if you see what it has on the surface, it has this, small hair-like structure. Very small, but they have this hair-like structure like this, right? Numerous, you will find numerous what? These are called cilia. What are these? These are called cilia. And what is this organism? This organism is paramecium. We are talking about paramecium. What paramecium has on its surface of its body? It has very small, numerous outgrowth, hair-like outgrowth. It looks like our eyelashes, small hair-like outgrowths. Okay, this is cilia. Now, if you take example of this, that is, looks like this, right? Like this. This is chlamydomonas, right? Chlamydomonas, if you see, this structure what you find here, these two. So these two are called what? These two are called flagella. These are called as flagella. Now what difference can you find out in these two? Yeah? What are you able to find out? Difference? Difference is that these are very small in size. These are long. These are very short in size. These are very long. Second difference is these are numerous in number. These are 1 or 2. Okay. So these are the main differences of cilia and flagella. Physically, if you see, structurally they both are same. But physically, if you see, they are quite long. They are quite small. And these are few in number. These are many in number. And what is the function of both? How they help to these cells? What they do? Their main function, both of their main function. If we talk about function, what is the function of cilia and flagella? Function is what? Function is it provides motility. It provides motility to the cells. Provides motility means it helps in movement. It helps these two cells to move. Now in NCERT they have given one particular this thing about cilia. Cilia, what it does, whenever it is present outside the body of any kind of organism, it helps in movement of that particular cell or organism. Okay, it beats in oar-like fashion. They have given this sentence. You need to remember that. It beats like oar-like fashion. Now, what is this oar-like fashion? That means oar-like movement it shows. That means whenever we are traveling in water with boats, okay, there will be some wooden structure which what we will do is we will use that and we will move the water back so that the boat moves front. Okay, whenever we want to move back, then we move the waterfront so that we move back. So this type of movement what we do with the help of that wooden that wooden thing is called oar. So the same type of movement it shows. It beats like that. It beats like that oar so that the cell moves forward. And what about this flagella? Usually the movement of flagella is not like this. Its movement is just like this. Like if you take a rope, somebody is holding it from one side, the other side what you will do is you will make this kind of movement up and down. So you will see that wave like motion of that. Such type of movement is movement of flagella. So flagella moves in that fashion, but cilia's movement is like oar. So this is you should remember in case they may ask this question, like oar like movement is shown by what? It's shown by cilia, not flagella. Flagella is wave like motion, you can say. The movement is like that. Okay, so I hope this is clear. Very best example if you want to talk about cilia and flagella, that means we need to relate it with ourselves, with human beings. Now whenever consider ourselves, okay, now let us see example in our body. So I hope you understood this much. Cilia is present in paramecium, the best example for that. For flagella, chlamydomonas, so many cells are there, right, which have this cilia and flagella. Now let us talk about some examples of our body itself, okay, of human beings, so that easily we can remember. So where you will find cilia, where you will find flagella. Let us consider for flagella. If you consider flagella, will you tell me what is this? Don't tell it is snake, okay? It looks like that. Anyways, I'm not drawing the particular shape of that, okay? Anyways, I'm just showing you. Head, neck and tail. If you see that, usually you will draw it like this, right? Like this you do. This is what I'm showing it in a microscopic view. If you see now, it looks like this, electron microscope. But usually it looks like this. So this is what? These both are what? These are nothing but human sperms. We're talking about sperms. If you see sperms, it has head, neck and tail region, right? And what is this tail? This tail is nothing but what? It is nothing but flagella, right? So it beats in that wave-like motion so that it moves forward. In human reproduction chapter, you are very well versed with the structure of sperm. There is mitochondria present in the middle here, this region, which gives energy for that, tail to move. With that ATPs, it moves, it beats. Because of the beating of that flagella, sperm moves forward. Now then, in human being, if you talk about cilia, where will you find it? Think about it, where you will find cilia? In case of human beings, if you talk, where you will find cilia? Cilia, if you consider in human beings, for example, in females, if you see, yes, this is a fallopian tube like this. I'm not drawing complete one, okay? And these are the ovaries like this. And you have this finger-like projections here, like this, fimbriae. And this is what? These two are fallopian tube. Usually when sperm enters the vagina, after that when it reaches, it needs to move till the egg. And where will be the egg present? Egg is present here in the isthmic junction, ampullary isthmic junction. If this is egg, then sperm needs to move here. For example, the sperm needs to move here inside. Now what makes this sperm move is the motility of sperm, that is the flagella. Now we are talking about cilia. So where is cilia here? If you see inside this fallopian tube, we have the small hair-like structure. It is completely filled with this small hair-like structure. Very small. As soon as the sperm reaches till here, that ciliary movement, that cilia moves like this. The movement of cilia will aid the sperm to move towards the egg. It's like taking that sperm towards the egg. It helps in movement. Whenever sperm comes till here, cilia beats in such fashion so that sperm should move and reach the egg. So these are what? The hair-like structures what you find here. So these hair-like structures, what we call them? We call them cilia. Why I took these two examples is, usually whenever we talk about ourselves, we remember this easily, and when we talk about some other organisms, we tend to forget it, right? So these are the examples of our own body, so we will remember this. Now in case of it is about males, this is about females, but both, like for males and females together, if you see where you will find cilia, you will find cilia even in tracheary, this one tube, trachea. In trachea we have small hair like structures. In order to repel or remove out the dust particles which has entered through our nose, so for that reason their cilia is present. Okay, many areas in our body we have this ciliary structures. Flagella, usually it is found in sperm. In sperm cell we'll find this flagella. I hope so you understood the examples. Fine. Now what we are talking about is mainly about the structure. Okay, fine. We know that these two are responsible for movement. It is helping in movement. Even here flagella is helping sperm to move. Cilia is helping sperm to move towards egg. And if you see other organisms, their cilia is helping in movement, like paramecium. Anyways, what we will do is now we are going to study the entire structure of cilia and flagella. Now if you see the structure of cilia and flagella, there is nothing much confusion here. We need to understand it clearly. If you have cleared with the previous chapter previous topic what we have done, cytoskeleton, in that also if we have done microtubules, if you remember that, then you will easily understand this. Okay, now cilia and flagella, I told you their number is different and the sizes, they differ in their size. But if you see the entire internal structure, it is similar. So whenever you see the cilia and flagella, they have this, they have this basal body. They have this basal body which is embedded within the plasma membrane. So this will be the plasma membrane like this of the cell. Okay. So this basal body is, it will be in this is plasma membrane. If I do this, you will understand, right? Fluid mosaic model. Okay. So basal body will be within the plasma membrane, embedded with that. And then it has a structure like this called hook, like this. Okay. And then what? And then it will have this filament like this. So this is basal body. This is called hook. And this is called filament. Now you people don't get confused with this structure of prokaryotic flagella. If you see this type of structure, both are same, but internal structure of prokaryotic flagella is different. We are talking about eukaryotic cilia and flagella. Okay, I hope so it's clear for you. So this structure is same. It has basal body, hook and filament, and it is embedded in plasma membrane, basal body, and then hook and filament are outside. But internal structure, if you see, it's different. Prokaryotic and eukaryotic both are different, and we are talking about which one? Eukaryotic flagella. Okay, now if I take a section of this anywhere from here till hook, if I take a section, cross section like this, yeah, if I take a section and if I show you here how it looks like this. If I cut it, right? If I cut it like this and I am showing this part now, it looks somewhat like this. Like this type of structure. This is this part and this section what I am showing you. So we are going to study about this, the internal structure. So here what you will see many microtubules here inside like this. And that arrangement we are talking about. Now if I rub this diagram, there is no problem, right? You are understanding what I am talking about. So now we will see the exact internal structure. In your textbooks they have given one internal structure. So what they have given is outside, this is a circular thing. not that perfect circle. Anyways, if you understand the concept, that's great. Okay, so this outer structure, what you find here, this one and this one is same. I'll show it with red only. Yeah, so this and this, what we're talking about, this is called plasma membrane, because it is covered by plasma membrane. So the outer structure what you find is called plasma membrane. Leaving this plasma membrane, whatever you have, the core, whatever inside core we have now, that core is called axoneme. That means leaving this plasma membrane, red color structure, inside whatever you have, everything is called axoneme. Clear? First thing is the outer layer is plasma membrane. Second, internal core, whatever we have, so many things are embedded in that. This is called axoneme. Okay? Now, next what you find here. See, this arrangement of cilia and flagella both, you know what we call? We call this arrangement as 9+2 arrangement. 9+2 arrangement. This is very, very important. Now, what is this 9+2 arrangement? What is this 9+2 arrangement? That's important, right? Okay. So 9+2 arrangement, they are talking about microtubules. Yesterday we have done microtubules, right? Microtubules will be made up of what? Which protein? Do you remember? Which protein? Microtubules are made up of tubulin protein. And how many microtubule will make one, protofilaments will make one microtubule? It is 13. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So this is one microtubule. Microtubule. These type of tubule you are going to see inside what I have shown here. But you will not see it like this. You will see another ring here. You will see another ring, but it is not of 13 microtubules. Even that is microtubule only, but it is not made up of 13 protofilaments. Here you will find 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So this is what? Both are what? Both are microtubules. One microtubule is complete with 13 protofilaments. The other microtubule is not complete, but it is attached to the first one and it is having 10 protofilaments. So what is this number? It has 13 protofilaments and it has 10 or sometimes 11, usually 10. So 13 and 10. These two are what? Togetherly, these two are microtubules only. But here we will not call it as one microtubule. Instead, we call it as doublet. This is called doublet. What it is called? It's called doublet. Doublet means what? 2 microtubules, but in a single structure. That is called doublet. Doublet microtubules, which is called doublet. One with 13, one with 10. Here I will not be able to show it clearly, so I have done it here only. So these things we are going to do it here. Okay, now I'll just show you with one like this. How many will be there? These type of doublets? 9. So 9 plus 2, no? We will see that. So this is 1 with 13, 10. एक हो गया. We will do one more here. 1 complete, 1 half. One doublet, 2 doublets. Okay, next one. 3 doublets. 4 doublets. Actually, it will be like this. Okay, then कितना हो गया? 4, 5, 6, 7, 8. One more we need to do. So what we will do? 1, 2, 3. Wait. 3, 4, 5, 6. 5, 1, 2, 3, 4, 5, 6. Okay. 7, 8, 9, and then like this. Not that good diagram, but please try to understand. We are talking about what? We are talking about these doublets here. Doublet is nothing but it is made up of two microtubules. One is having 13 protofilaments, the other one is attached to it, and it is made up of 10 micro protofilaments. Then if you see these doublets, how many are there? 1, 2, 3, 4, 5, 6, 7, 8, 9. So that is why we have given here 9. And then again we have 2, no? 9 are doublets. What about the 2? Those 2 are found in between here, like this. And they are not doublets, they are singlet. That means they have what? They have 13 protofilaments. This one, if you see, only one structure is found here, one structure is here. So how many, if you talk about 9 plus 2 arrangement, 9 doublets are there and 2 singlets are there. And how the singlet looks? Same thing. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. This is a singlet. Singlet is having how much? It has 13 what? Protofilaments. All these things are what? Protofilaments, right? Protofilaments. And please, if you are watching this video without watching the previous one, it's of no use at all. Please go back, search it, find it out, study that, and then come here. Okay? So how many doublets are there? 9 doublets. How many singlets? 2 singlets. So what is, so 9, we are not considering this 2 as 2. We are considering this 2 as 1, because it is not 13 and 13, it is 13 and 10. Here it is 13 separately, 13, 13. So we consider it as singlet, and there are 2 singlets. So it is 2. 2 singlets. How many doublets? 9 doublets. Is this clear? Okay. Outside, they all are present outside. These outside microtubules, what you have, you're understanding, no? These tubes, you know, these tubules, you know, they are like this. Continuous tubules there. You know, they are sheet. Yesterday I have shown you. They are continuous. I cannot show like that. Show in book also they have shown the section. I am showing you this section like this. This means these things are continuous till here. They are continuous. Okay. I hope you people are understanding. So outside it is covered by plasma membrane. Inside the core is called axoneme. In the core axoneme, so many things are there. What are there? 9 doublet microtubules are there. Within that, inside, in the core, exactly in the middle, there are 2 singlets. Now there is a bridge which connects these 2. There is a bridge which connects these 2. They have shown in the book. This bridge is called central bridge. We call it as central bridge. What it is called? Central bridge. What is the function of central bridge? It is joining the two microtubules which are present at the center, which are singlets. Singlet microtubules. There are 2 which are connected by a central bridge. Then, like plasma membrane only, these are covered by some other membrane, and we call it as, like this they have shown. You can show it continuous also, but in book they have shown like this only. This is called central sheath. What do we call them? We call it as central sheath. S-H-E-A-T-H. I hope you people are able to see. Okay. So this is called central bridge, which connects 2 microtubules which are at the center, singlets. And the sheath which covers it, it is called central sheath. I hope this much is clear. 1, 2, 3, 4. Shall I rub these things so that you can understand still more clear? Take down this much. I hope now you people are able to see it. I'm trying to zoom it out to show you the labelings. My board is very small, so please try to adjust. Okay, I'll wrap these things. So I hope you have understood what is a doublet and what is a singlet. I'll wrap these things now. Okay, this much is clear. Now what you will see is that next thing. Now these central sheath, there is a connection with this outer ones and inner ones. These two are central microtubules. These are outer ones. So these microtubules which are present outside, we call them peripheral. We call them with naming we will do with black. Okay, you will see it clearly. This is axoneme, I told you, the core. I'll write that also, you will understand that. The core of the inside core is called axoneme, and then these doublets which are outside, we call them peripheral. Peripheral what? Doublets. Peripheral microtubules, which are 9, right? They are 9. And what? They are doublets. Everything I made it clear. Peripheral microtubules are 9 and they are 9 doublets. They are found in doublets. Then these are internal microtubules which are singlets. That also if you want you can write. These are internal microtubules and these are what? How many are there? 2 are there and 2 are singlets. This much is clear. Now this is called central sheath. That also is clear now. Now these things, this central sheath is connected to all the 9 what? All the which color shall I use now? I'll use blue only. So these central sheath, you know, it is connected with this peripheral microtubules. With what? Like this. And it is connected to the one which is complete circle. Okay, remember that. Like this. This is one connection. This is one connection. Connected here also. Connected here also. Here. Here. This. This and this. Okay, I think I made a mistake over here. Complete circle, you know, that will be connected. Everywhere I think I am shown with complete circle. So these lines which I have drawn, which is connected with this peripheral microtubule to the central sheath. This is called what? This is called radial spoke. What do we call them? We call it as radial spoke. What is the function of radial spoke? Radial spoke connects the peripheral microtubule to the central sheath, and it is connected to the complete 13 protofilaments structure irtadalla. That is called one singlet. I mean in doublet we have this complete structure. So that is connected with this sheath. With what? With radial spokes. Then how many radial spokes will have? If you are having 9 doublets, obviously how many radial spokes will have? Will have 9 radial spokes. Clear? Then what we need to study here? After this, we have only few things remaining. Now these things are connected. There is a connection with these things. I'll show it with black. They have a connection like this. Like this. Are you able to understand? The doublets are also connected. Like this. I hope it is very messy anyways. I think you're understanding also. So these doublets are connected by this black structure what I am showing. These things are called inter doublet bridge. Inter doublet bridge. How many bridge we have studied here? Central one is called central bridge. Doublets are connected with doublet, inter doublet bridge. And then peripheral microtubules are connected to central sheath by radial spokes. These 3 things clear. Last what we are remaining with is there are some structures like this. If you have seen, you know, like, okay, you will see like these type of structures, no? Internal, like this, like this. Everywhere it is there, and it is seen with complete one only, like this, no? Somewhere it is 3 outside, 2 inside, I think. Okay. These are called Dynein arms. In NCERT, nothing is given about this structure, but it is very, very important. Without these, no movement of flagella or cilia, like this. So this complete structure is what you see like this. So these things are called Dynein arms. Okay, no place to write about that. I'll rub this. What is this called? You people know that I am talking about structure of cilia and flagella, right? So here, these black things what we have done, this is called a Dynein arm. Outside it will be 2 or 3, inside it will be 2. These are outer Dynein arms. Inner side also they are there, no? These are inner Dynein arms. These are called Dynein arms. In NCERT nothing is given about this Dynein arms. But see, what is the function of Dynein arm? Can you see they are connected to what? They are connected to microtubules. Whenever there is movement, whenever movement has to happen, that movement actually happens because of this Dynein arm. These Dynein arms will get attached to the microtubules which are in the further. Now these Dynein arms, these Dynein arms which are there, these ones, these will connect to this one. They get attached and then they move. Are you able to see here how they are connected? So these Dynein arms, you know, if you see, they all are connected like this for the other ones. Like this. Like this. What they do is, whenever their movement is required, they touch the further filaments, microtubules, and move it like this. Up and down movement is done with the help of ATPase. You know ATPase, ATP synthase. What it does, it forms ATP. With the help of energy, when ATP is produced, that ATP is used and this movement is done. Dynein arms will move, they touch the other ones in the front, the microtubules, and it moves like this, up and down motion. And because of that, what will happen? You know, there is a movement for this whole cilia or flagella. So this Dynein arms, they have this ATPase activity, so that it uses this ATPs and they help in, they get attached and detached from this microtubules which are present here. So inside these two are not connected with any kind of Dynein arms. Only outside you will find it. Okay, so this is not given in NCERT. Anyways, you need to know that. So I hope the structure of these things are clear. Anything if I have left, let me know. I hope I have done completely. So once again, I'm just going to revise it. First, it is plasma membrane. Inner, the core is called axoneme core. And then they have 9 doublets, 2 singlets. Okay, how many? Finally, I'm writing it. 9 doublets and 2 singlets. Doublets means what? Doublets, they are nothing but like this. They are made up of how many? I'm not doing correctly, but 13, and the other one will be 10. Singlets will be what? Completely they are made up of how many? 13 protofilaments. Now it is clear. Now outside there are 9 doublets. Those are called as peripheral doublets, peripheral microtubules, which are doublets and they are 9. Inside we have only 2. They are internal microtubules and they are only 2. They are called singlets. Now these 2 singlets are connected by a bridge called central bridge. Outside to this tubules you will find a sheath covered, and that is called central sheath. It's a proteinaceous sheath again. After that, this 9 tubules which are found outside, doublets, these are connected by this bridge again, and this bridge is called interdoublet bridge. And then what you will find is there are arms which comes outside from each microtubule, and those are called Dynein arms, which are towards the plasma membrane. They are outer Dynein arms, which are towards the central sheath is inner Dynein arms. Finally, the function of Dynein arm is to attach to microtubule, move it. Whenever movement is not required, leave it. Clear? Because of that, there is a movement in this flagella. Ultimately, flagella and cilia move, and then the organism moves. Clear? This is the internal structure of cilia and flagella. And what we need to study next is centriole, centrosome. So there you should not get confused. So please be perfect with this. Same type of structure you're going to see there also, but it is quite different. If you are well versed with this, in NCERT they have given this diagram, and in centriole and everything, in written the structure is given there also. So both the structure we need to do. Okay, please be clear with this. Any doubts, please comment. Okay, thank you.